Welcome to today's video, in which I'll be discussing recent developments in rapid microbial methods with Dr. Tim Sandle, the Head of Microbiology, Risk Management and Sterility Assurance at Bioproducts Laboratory Limited. Hi Tim, thanks for joining me today.
Hello, thanks for having me.
No problem at all. So let's make a start. Can you first tell me how rapid microbial methods are different to traditional microbial techniques and why they're becoming more popular?
Okay, I think you can define rapid microbiological methods in two ways. One is being rapid, which is all about the time to result. They're also often alternative to what's in the compendia. So in that's a sense of offering improved detection and greater sensitivity. And they also have elements like automation and the ability to capture data digitally. But I guess the two fundamentals are accuracy and then with obtaining the desired information in a shorter time period.
And obviously the FDA recently introduced process analytical testing. So has this played a role in their adoption?
Yes, I think it's helped kind of nudge the industry in that direction. So process analytical technology is kind of centered around designing, analyzing data in real time in manufacturing environments so that those people working as part of the manufacturing process can collect information and also make decisions based on that and address things like quality. And here data is directly connected to the process and can also, in some cases, be directly inputted into the batch record, for example, through connecting equipment up. And this includes both chemical and microbiological data. And I think the FDA like it because they can make real-time risk-based decisions.
What are some of the different types of rapid microbial methods in use in the pharmaceutical industry and why are they used? What are their applications?
Okay, so I think we can have, you divide rapid methods up into kind of different ways. So we have like qualitative rapid methods, which are kind of looking for like presence and absence. So this could be looking for an organism like E. coli in a water system, for example. And then we have qualitative methods, which are trying to enumerate, trying to count how many bacteria might be present in a sample. And then we have like quantitative methods looking at potency or toxicity, like say the level of endotoxin in a water system. And then we have identification-based methods, which are trying to tell us what species of microorganism it might be, say based on the phenotype or genotype of the organism. So some of these methods are growth-based, so we're looking for some kind of detection, some kind of signal as the microorganism is growing. Then we have some which are like viability-based, so perhaps we're looking at, say, fluorescence staining, or we're using laser excitation. So that here there's no need for the microorganism to be growing, it just needs to be present. Okay. And then another category where we're looking for some kind of cellular marker, like looking for ATP, for example.
Okay, fabulous. And what are some of the most recent developments in the field? How have things been changing?
I think there's a number of different changes. I mean, if we take the pharmaceutical context, then there's been advancements with optical spectrophotometry, which is using like light scattering with different sensors. And this is designed to say, we might have particles in the air. How many of those particles might be biological and then by inference microorganisms? That helps with contamination control. And that technology is quite exciting for looking at like grade A environments and aseptic filling, for example. Then there's been some developments with headspace analysis. So this is looking for the presence of carbon dioxide, say, in bottles from an aseptic media film. So it's a way of more defensively saying how many of those are likely to have microorganisms that are growing in them. Then there's some technologies where you basically get a rapid result because you're looking for micro colonies, which you can't yet see with the naked eye, but you can use like a blue light laser excitation to try and detect those colonies before they become visible. So you can react to that data far more quickly. And then I guess another final example would be the advances in the kind of lab on a chip technology, which are basically miniaturized test platforms with different reagents and substrates. And here you can do things like, if you're looking for a new antibiotic, you can do mass screening of different compounds far more quickly and far more to scale than would be possible with the kind of more traditional methods of doing that. So there's like 4 examples of quite interesting and innovative technology.
Fabulous. And has COVID had an impact on rapid microbial methods and their implementation? Obviously, it's introduced digitalization on a mass scale, but has that applied here?
I think it's shown the importance of having them because some of the way that the COVID vaccines have been turned around has been really, really fast. And that's shown that the faster we can get laboratory results out, the better. The one area I think I most understand with the development of the COVID vaccines is with the need to screen for endotoxin. Because obviously these vaccines are being injected into people, we don't want to have high levels of endotoxin. So I think some of the advances that have taken place more recently with recombinant lysates and with the use of automation and faster endotoxin test kits has really helped with the vaccine production. But in general terms, you know, if we have another pandemic, a different virus in the future, to be able to get drugs out more quickly, we want to be able to ascertain the quality of those drugs more quickly. So rapid methods are key to that process.
Fabulous. So what are you particularly excited about in the field of rapid microbial methods? What gets you to the lab in the morning?
Yeah, I think the gold standard is always going to be the rapid sterility test. And there's been some advances and in some sort of vaccines and cell culture work that's coming online, but that's the real one. To get that for mainstream pharmaceutical products and get that established, that's really going to pay benefits because it'll be more accurate and sensitive, and we'll know whether we've got a problem more quickly. In terms of like more immediate technologies, then I think it is the spectrophotometric counters to do this distinguish between non-viable particles and biological entities. And you know, as that technology matures, having those embedded into filling lines, for example, so we know the risk as we're filling medicinal products. And I think, you know, having more indication with water systems, you know, a lot of pharmaceutical water systems are very big, very complex, and be able to put on instruments, say based on flow cytometry, to give an indication that the microbial levels are rising, and then say automatically shut down that water system to prevent it being used in the production of a product. That's quite interesting. And then the technologies that allow us to do more accurate plate counting, because another area that comes into play there is data integrity, because distinguishing colonies on an agar plate, for example, isn't always the easiest thing under different types of light or different eyesight and different people. So being able to fully automate that with digital scanning technology is pretty awesome as well.
It is quite amazing what we're beginning to achieve with automation and technology. But tell me, are there any limitations for rapid microbial methods at the moment?
I think there is in different levels, really. So first of all, I think there's you know, thinking about what we want to do in advance, and some rapid methods produce the result more quickly once the sample prep's been done, but there may be limitations on throughput. So the laboratory needs to consider how long does it take to prepare the sample, and also whether the laboratory's got time to validate it as well, because you know, there's often too much equipment that's purchased and then stacks up. But perhaps more fundamental with the non-growth based methods, then where we're using a non-culture method, often the rapid method provides something in a different unit. So it might be a degree of fluorescence, for example. And it's not always clear how that translates back to the actual number of microbial cells, or what might be the equivalent to a colony forming unit. There's also cost issues. These aren't the cheapest instruments to buy. And it's also really important to make sure the validation's good, particularly what's called robustness, which is testing out the instrument with different analysts and different reagents and really showing that it can do the job it needs to do when it gets into the workplace.
What are some of the key limitations or key considerations when implementing rapid microbial methods?
I think it's important to sometimes not to get too wowed by the technology and the sales rep as well. So you know, a lot of the rapid methods that are offered are sometimes in earlier stages of development. So I think it's important to sort of say, is this technology mature, and is there good supporting literature? You know, has someone written about it in European Pharmaceutical Review, for example? And then I think it's really important to follow the guidance. And there's really good chapters in the European Pharmacopoeia, which is 516, and in the United States Pharmacopoeia, which is chapter 1223. And they give really good guidance and a step-by-step approach and things. But it's really important to always start with risk assessment to work out, you know, what we're trying to do and what might go wrong, to follow the classic validation steps of design, installation, operational and performance qualifications. To be careful if we're going to compare the new rapid method with a conventional rapid method, because we could be looking for very different things, and doing those comparisons can often be quite fiddly. And I think, you know, we're going to get scrutinised by a regulator when we put the request in. So we want to make sure that, you know, we've done it with the sample size is appropriate, and all the variations of taking different samples, testing at different times of day, and really kind of testing out that system. We've got a good data set that we've appropriately analysed, and, you know, we can stand up and stand by the data, essentially.
Of course.
I think just one of the myths is sometimes that regulators are kind of a brick wall to rapid methods, and in my experience that's not true. It's actually the reverse, is that regulators are actually trying to encourage us to look at rapid methods, and some of that is inherent in the revision to Annex 1 that's coming out. So within there, for example, it looks like the limit for grade A environmental monitoring, for example, is going to change from 1 CFU to no growth. And by stating no growth, that's a nudge that some rapid methods can be considered. So I think, you know, there is general encouragement in investing in technologies that ultimately help to protect the patient.
Fabulous. Well, that's all we've got time for today. So thank you, Tim, for your insights into rapid microbial methods. We hope the audience enjoyed this video. Please let us know your thoughts in the comments section at the bottom of our webpage, and I hope you'll join us for the next one. Thanks for your time for now, and goodbye.
Bye.